Welcome to BioBits and Bytes. Okay, let's unpack this. Imagine having like an incredibly precise dimmer switch for every single gene in a cell, not a crude on-off switch that cuts the DNA, but something that can gently, reversibly, and dynamically turn gene activity up or down. Have you ever considered what completely new avenues this opens for understanding disease, developing therapies, or even engineering entirely new biological functions without ever permanently altering the core genetic blueprint? Think of it like being able to fine-tune the performance of a complex machine, maybe not by replacing its core components, but by adjusting its internal programming on the fly.
That's a powerful way to frame it, a really good analogy. Today we're taking a deep dive into some truly revolutionary tools in genetics. CRISPR activation, which, we'll call CRISPRa, and CRISPR interference, or CRISPRi.
Yeah.
For many people, when they hear CRISPR, they think gene editing, the cutting of DNA. But these systems are fundamentally different. They represent a new frontier in gene regulation, offering remarkably precise ways to modulate gene expression. That is, to control exactly how much or how little a gene is read and translated into proteins. It's about orchestrating the symphony of life, sort of adjusting the volume and tempo of individual instruments rather than rewriting the entire score.
Okay, so our mission for this deep dive is to guide you through this fascinating landscape. You want to demystify exactly how CRISPR and CRISPR operate at a molecular level, crucially, without altering the DNA itself. Then we'll dive into their incredibly diverse and impactful applications, spanning from fundamental biological research and the engineering of new metabolic pathways to truly transformative potential in human therapeutics.
Yeah, absolutely.
And finally, we'll grapple with the current challenges facing these technologies, particularly in less studied organisms and, in clinical translation, before casting our gaze to the exciting future directions and groundbreaking innovations emerging in this space.
And to help us navigate this complex but really rewarding topic, we've pulled insights from a stack of leading research. Our sources include a comprehensive PubMed review titled Advances in CRISPR-Cas Systems for Epigenetics, which gives a great overview of the DCAS9 Foundation. We'll also draw from a detailed Wikipedia entry on CRISPR activation for some foundational concepts, plus a highly informative Frontiers mini-review on CRISPR-based approaches for gene regulation in non-model bacteria, which is critical for understanding applications outside of, the usual lab organisms.
Right, beyond E. coli.
Exactly. And finally, we have a very insightful PMC article on CRISPR-based therapeutics, which really grounds our discussion in real-world clinical potential and, importantly, the challenges. So these sources provide a pretty robust foundation for understanding both the how and the why of these revolutionary tools.
Okay, perfect. So let's start at the beginning then. Before we jump into these dimmer switch versions, let's quickly ground ourselves in the foundational CRISPR-Cas9 system itself. We hear about it constantly in the news, usually in the context of gene editing or gene cutting. But what exactly is it at its most basic level, and where did this extraordinary molecular machinery actually originate?
Right. Well, at its heart, CRISPR-Cas originates as a form of prokaryotic immunity. This isn't something humans invented. It's a naturally evolved, ancient bacterial defense system against invading viruses or bacteriophages and foreign plasmids. You can sort of imagine a bacterial immune system that actually remembers past infections. It comprises one or more CRISPR-associated, or Cas, proteins, these are basically the molecular scissors, and a short guide RNA, or gRNA. Now, this gRNA is the key to its precision. It's a small RNA molecule that contains a sequence complementary to a specific target sequence found in the foreign DNA or RNA of the invader. So the gRNA literally leads the Cas protein, typically Cas9, with remarkable accuracy, directly to that target sequence through complementary base pairing. It's like a perfectly matched lock and key. Originally in its natural state and in most gene editing applications, the Cas protein would then cleave or cut that foreign molecule, often after recognizing a specific short sequence called a protospacer adjacent motif, or PAM sequence, which acts kind of like a molecular flag right next to the target site. This cut effectively neutralizes the threat, like a bacterial shredder for viral genomes. So in essence, it's a sophisticated search and destroy system.
But here's where it gets really interesting, right? And where CRISPR and CRISPRi diverge from that search and destroy function. With these systems, we're talking about not cutting the DNA. So how do they achieve that remarkable trick? What's the fundamental key modification that allows them to dial gene expression up or down without making a permanent, irreversible cut to the genetic code?
The secret lies in a truly ingenious modification of the Cas9 protein, creating what's known as DCAS9, which stands for catalytically inactive Cas9. Researchers achieved this by introducing specific point mutations into the Cas9 gene. The two most common and effective mutations are D10A and H840A. These are specific amino acid substitutions. D10A means aspartic acid at position 10 is changed to alanine, and H840A means histidine at 840 is changed to alanine. These particular amino acids are absolutely critical, almost like the cutting edges, in the enzyme's two distinct nucleus domains, RuVC and HNH. They normally coordinate magnesium ions essential for DNA cleavage. By strategically changing them, researchers effectively created a Cas9 that still possesses its incredible ability to recognize and bind precisely to a target DNA sequence, thanks to its gRNA map. But its catalytic jaws are rendered completely inert, incapable of making a cut.
Okay, let's pause and simplify that part again because it's so fundamental to understanding these technologies. So DCAS9 acts as a highly specific GPS for the genome, taking the gRNA exactly to the right DNA address, but without causing any damage. It just sits there, a precise, inert placeholder. It's essentially a DNA-binding protein on demand.
Precisely. Exactly that. And this ingenious property, this ability to bind specific DNA sequences without cutting them, is what synthetic biologists have powerfully leveraged. Instead of using Cas9 for permanent gene editing, they repurposed DCAS9 to build entirely new tools that modulate gene expression, creating a whole new level of control over cellular processes. It really opened up a world of reversible, tunable genetic intervention.
Okay, so DCAS9 is our GPS, guiding us to the exact gene we want to influence. But once it gets there, how do we actually turn genes up or down? What's attached to this DCAS9, or, what does it recruit that allows it to exert influence over gene expression after it finds its spot?
Right, that's where transcriptional effector domains come in. Think of these as modular tags or functional units that could be either directly fused to the DCAS9 protein or recruited to the DCAS9 complex by modified guide RNAs.
Sure.
These effectors are the actual dimmer switches. So for CRISPR activation, or CRISPRa, the DCAS9 is typically fused with or recruits specific transcriptional activators. These are proteins or protein domains that boost a gene's transcription. Common examples include VP64, that's a potent artificial activation domain from a virus, the P65 domain of NF-key, or the Epstein-Barr virus, or transactivator, or NADA. What's even more potent and widely used today are systems like the CRISPR Synergistic Activation Mediator, or SAM complex. The system goes beyond just one activator. It's brilliantly engineered to recruit multiple unique activator domains to a gene's promoter simultaneously. In the SAM system, VP64 is directly fused to the DCAS9. But crucially, the gRNA itself is engineered with RNA aptamers, specific RNA sequences that act as binding sites. These aptamers then recruit two other distinct transcriptional activator proteins, MS2P65HSF1 and MCPVP64, to the DCAS9 gRNA complex. Together, this multi-activator complex dramatically boosts the gene's transcription, often achieving hundreds or even thousands of times more expression than basal levels. It's like having a whole team of molecular cheerleaders all shouting at the gene to start expressing itself.
Wow. Okay. Multi-pronged attack.
Exactly. And now, for CRISPR interference, or CRISPRi, it's the exact opposite mechanism designed to turn genes down. Here, DCAS9 is typically fused with a powerful transcriptional repressor domain. The most commonly used one, and highly effective, is the Cripple-associated box, or KRAB domain, sourced from a human protein. This DCAS9 KRAB complex interferes with gene expression in a few crucial ways. Its primary mode of action is, well, partly acting as a molecular roadblock, but more significantly, it recruits histone deacetylase, HDAs, and other chromatin-modifying enzymes. These enzymes then compact the local DNA, converting open, accessible chromatin into a more condensed, transcriptionally repressive state. It's like rolling up a blueprint so tightly that the cellular construction crew can't even unroll it, let alone read it. So, while it can physically block RNA polymerase from binding or extending, its dominant effect is often through inducing this repressive chromatin state, creating a more durable and potent gene silencing effect. In some specific cases, it can also cause RNA cleavage, but its main power lies in blocking transcription initiation and promoting that repressive chromatin.
Okay, so the guide RNA, the gRNA, is clearly crucial for this precise targeting. But is designing gRNAs for CRISPR and CRISPRi the same as designing them for traditional CRISPR knockout, where you're trying to permanently disable a gene? It seems intuitive that you'd always just target the start of the gene, right?
No, exactly. And this is a really critical distinction for anyone looking to implement these tools effectively. It's not quite that simple. While traditional CRISPR knockout often targets early exons in protein-coding genes, the idea being to create frame shifts and prevent expression of a functional protein, CRISPR and CRISPRi have very specific optimal targeting windows relative to the transcriptional start site, or TSS, of a gene. And this isn't just a slight preference, it's a major factor in how well they work. For CRISPR, especially when using the dCas9K fusion. The most efficient gRNAs are designed to target within the first 100 base pairs downstream of the TSS. However, the effective window for potent repression generally spans from about 50 base pairs upstream to maybe 300 base pairs downstream of the TSS. Targeting too far downstream quickly loses efficacy. This strategic placement helps the dCas9 complex effectively block RNA polymerase or induce local chromatin condensation right where transcription needs to initiate or elongate.
Right at the gate.
Precisely. Now for CRISPRa, the optimal region for gRNA binding is quite different. It's typically located upstream of the TSS. Specifically, the strongest activation is achieved when gRNAs target within the window from about -400 to -50 base pairs relative to the TSS. This applies even to those powerful multi-activator systems like SAM. Targeting outside of this region, particularly too close to or within the coding sequence, significantly reduces or even abolishes activation.
That's fascinating. Have you ever wondered why these optimal targeting windows are so distinct for activation versus repression? It seems counterintuitive that you wouldn't target the same spot if you're trying to influence the same gene. It feels like we're interacting with the gene at fundamentally different moments in its activation cycle, and I'd love to understand the why behind that.
That's a great question. Let's break it down. You've hit on the core molecular choreography there. It absolutely relates to the fundamental mechanism of how each system works to influence the transcription machinery. Repression, with CRISPR, can often occur by simply physically blocking the RNA polymerase after it attempts to start transcription, or even by blocking its elongation. So targeting a region just after or slightly into the gene's coding region is effective for physically interfering with the movement of the transcription complex or compacting the DNA in that immediate vicinity. The dCas9 acts like a gatekeeper right at the entrance, or just inside, basically.
Okay, makes sense.
Activation, on the other hand, with CRISPRa, needs to bring the transcriptional machinery to the promoter region to initiate transcription efficiently. Think of the promoter as the gene's on switch. Targeting upstream of the TSS positions the activators to recruit all the necessary basal transcription factors and RNA polymerase that kickstart the gene's expression. It's like standing outside the factory, ringing the doorbell and inviting the workers in, rather than standing inside trying to block the assembly line. Different goals require different strategic locations.
Right, recruiting versus blocking.
Exactly. Beyond these optimal windows, there are also practical challenges in designing highly effective gRNAs. Factors that can significantly reduce efficacy include gRNAs with very long protospacer lengths, generally over 21 base pairs. Also, the presence of nucleotide homopolymers, like a repeating string of AAA or GGG within the protospacer region, can make the gRNA less effective or may be prone to off-target binding. And significantly, the chromatin environment around the targeted genomic region can limit the dCas9 complex's access to the site. If the DNA is tightly wound up and inaccessible, even a perfectly designed gRNA might struggle to bind effectively.
So accessibility is key.
Absolutely. For large-scale screens, especially genome-scale ones where you're testing thousands of genes, it's vital to have highly active gRNAs to ensure robust results and avoid false negatives. This is why most pooled gRNA libraries often include multiple gRNAs per gene, typically between 3 to 10, to increase the likelihood of achieving robust activation or repression. Pooled gRNA screens are particularly useful for empirically optimizing gRNA activity and informing prediction algorithms. They allow researchers to test thousands of gRNAs with relatively minimal effort, helping to clarify how various factors like sequence, position, and accessibility influence gRNA activity in vivo. This has led to the development of highly active and more compact gRNA libraries, which in turn reduces the number of cells needed for genome-scale screens, making them more feasible and cost-effective. It's sort of an iterative process of design, test, and refine.
Okay, that makes sense for optimizing the guides. Now let's talk comparisons. Before CRISPR came along and really revolutionized gene silencing, RNA interference, or RNAi, was a go-to method for many researchers. How do these two methods stack up against each other? What are the key differences a researcher needs to consider when choosing between them? And what makes CRISPR, often a superior choice?
Yeah, that's a great question, and it highlights the constant evolution of molecular tools. CRISPR offers some really significant advantages over RNA, which, while revolutionary in its time, definitely has inherent limitations. So RNAi works in the cytoplasm by triggering the degradation of messenger RNA, or mRNA, preventing it from being translated into protein. The cytoplasmic location of its endogenous machinery makes it less effective for targeting nuclear RNAs like long non-coding RNAs or pre-mRNAs, which we're increasingly understanding play crucial roles in cellular processes and disease. So if your target isn't a cytoplasmic mRNA, RNAi might just fall short. Additionally, RNAi can cause significant off-target effects. The short interfering RNAs, siRNAs, or short hairpin RNAs, shRNAs, used in RNAi can bind to unintended, partially complementary mRNA sequences, leading to unintended repression of other genes. This can also happen by interfering with endogenous small RNAs, like microRNAs, by displacing them from the RNA-induced silencing complex, or RASC. This leads to what we call off-target phenotypes results that aren't directly related to your intended gene knockdown, which can really muddy your experimental results and lead to false positives.
Making interpretation tricky.
Very tricky. CRISPRi, in stark contrast, largely avoids these issues. Its components are entirely exogenous, meaning they're introduced from outside the cell, and crucially, it operates directly on the DNA in the nucleus, not just the RNA in the cytoplasm. So it can target nuclear non-coding RNAs effectively, and its precise DNA-level binding largely eliminates the off-target issues seen with RNAi's sometimes promiscuous RNA binding. In large-scale screens, studies have consistently shown that CRISPR generally provides more robust phenotypes, stronger, more consistent effects, and significantly fewer off-target effects than RNAi. This leads to much higher signal-to-noise ratios and greater reproducibility. Essentially, CRISPR is a cleaner, more precise, and more robust tool for gene silencing, giving researchers far greater confidence in their findings.
That makes perfect sense for precision. And what about comparing CRISPR to its original cutting cousin, traditional CRISPR knockout, or CRISPR-Co? It seems like if you want to get rid of a gene's function, a full knockout, a permanent removal, would be the most straightforward and definitive approach. What are the situations where CRISPR, with its temporary repression, is actually preferred over a permanent cut?
Well, while CRISPR-Co is undoubtedly powerful for permanent, irreversible gene inactivation, it certainly has its drawbacks, particularly in certain experimental or therapeutic contexts. The double-strand breaks that traditional Cas9 introduces can induce cytotoxicity and genomic instability. This is especially problematic in delicate cell types, or in cells that are difficult to transfect, or, say, in cancer cells that already have highly amplified and unstable genomic regions. Moreover, it's not always a clean knockout. About a third of the random insertions or deletions, called indels, that it introduces during DNA repair might not actually lead to a complete knockout or a frame shift. This means the targeted gene might retain some partial function, leading to incomplete or variable phenotypes.
Ah, okay, so it's not always a clean switch off.
Not always. Furthermore, targeting non-coding regions, which we now know are hugely important for regulation, is harder with CRISPR-Co. It typically requires 2 cuts to create a sufficiently large mutation for a loss of function, which can be inefficient. But crucially, and this is where CRISPR truly shines, for essential genes, genes that are absolute vital for cell survival, a complete knockout can be lethal. If you knock out an essential gene, the cell dies, making it impossible to study its partial roles, the nuances of its function, or how it contributes to cellular processes beyond just survival.
Right, you can't study a dead cell.
Exactly. This is the sweet spot for CRISPR. Because CRISPR avoids DNA cutting altogether, it bypasses these issues. It doesn't cause DNA damage or genomic instability. This makes it an ideal tool for perturbing non-coding genes, or for applications where you need a reversible or titratable gene knockdown. You can dial down the expression of an essential gene just enough to observe its effects without killing the cell. It's a superior choice for investigating essential genes where a partial, tunable knockdown is desired, allowing researchers to explore a range of expression levels and uncover subtle phenotypes that a lethal knockout would obscure. It offers a much finer resolution of gene function.
That's a very practical advantage. Now let's flip to the gain-of-function side. How does CRISPR compare to simply overexpressing an open reading frame, or ORF, which has been a common method for turning gene function up? Again, it seems like just flooding the cell with the gene's product would be the most direct way to study its effects.
You're right. ORF overexpression has been a workhorse in biology for decades. But CRISPR offers several key benefits that address some of its inherent limitations. Traditional ORF overexpression often uses strong, constitutively active viral promoters like the CMV promoter, which are designed to drive very high levels of gene expression. This often leads to unnaturally high, supraphysiological expression levels. While this can be useful for some applications, it might not accurately reflect how a gene functions under normal physiological conditions. It could potentially lead to misleading results or even cellular toxicity from just too much protein. It's kind of like trying to understand a car's performance by only driving it at maximum speed, right?
Yeah, not very realistic.
Exactly. CRISPRa, by directly targeting endogenous gene promoters, the cell's own natural regulatory elements, is much better suited for achieving more physiological or near physiological expression levels. You're essentially boosting the cell's existing control system, leading to a more natural upregulation. This also means that CRISPR is more likely to upregulate the most relevant splice variants of a gene, which might be unknown or difficult to isolate and clone in traditional overexpression methods. When you're using the native promoter, you're more likely to get the full, correct suite of products as regulated by the cell itself. Plus, from a practical standpoint, building genome-scale CRISPR libraries is generally easier and less resource-intensive than creating ORF libraries of similar scale. You're not dealing with cloning thousands of entire coding sequences. You're just designing relatively short guide RNAs.
Much simpler on the library construction side.
Much simpler. Overall, when you consider all these tools together, CRISPR-Co, CRISPR, and CRISPRa, they provide incredibly complementary insights into gene function. While CRISPR-Co offers a binary, permanent outcome, the gene is either off, permanently broken, or largely unaffected. CRISPR and CRISPRa allow for reversible, titratable gene expression over a broad dynamic range, sometimes exceeding 1,000-fold. This flexibility is incredibly valuable. We've seen studies that identified both activators like SBI1 and repressors like GANA1 as key regulators of cell growth in parallel CRISPR and CRISPR screens. In that specific example, researchers saw that both activation of SBI1 and repression of GATA1 increased cell growth in K562 cells, which nicely confirmed SBI1's known inhibitory effects on GATA1 activity. Used together, these tools greatly strengthen findings and provide a much more detailed and nuanced understanding of complex gene networks and biological processes. They allow us to move beyond just asking, what does this gene do, to how much of this gene do we need for X effect, and how does it interact with Y in a dynamic cellular environment. It's a leap from simple observation to precise cellular engineering.
That flexibility sounds like a true game changer for basic science. So let's explore some concrete examples. How are researchers actually putting these powerful tools to work in the lab right now? Let's start with mapping gene function.
Yeah, one of the most common and foundational applications is indeed mapping gene function on a large scale. This is where scientists systematically alter a gene's expression, either increasing it with CRISPR or decreasing it with CRISPR, and then carefully observe the resulting changes in cellular behavior, phenotype, or even disease progression under specific experimental conditions. It's essentially reverse genetics on a grand scale. CRISPR, as we discussed, is particularly powerful for investigating essential genes, those genes a cell absolutely needs to survive. Since its repression can be finely titrated, scientists can achieve a partial knockdown, preventing complete cell death. This allows them to study the subtle, dose-dependent effects of these vital genes, something that's impossible with a lethal full knockout. This has been invaluable for understanding the precise contributions of genes to fundamental processes like cell division, metabolism, or stress response.
Uncovering those non-lethal roles.
Exactly. CRISPR, on the other hand, is a fantastic tool for activating silent genes. These are genes that aren't typically active under normal lab conditions, or might be completely dormant in a particular cell type. By activating them, researchers can uncover their functions and the products they create, which might include novel proteins, non-coding RNAs, or even entire biosynthetic pathways. This is particularly exciting in microbiology, where CRISPR can be used to activate entire silent biosynthetic gene clusters in bacteria. These clusters often encode for the production of secondary metabolites like antibiotics, antifungals, or other valuable natural products, which are typically only expressed under very specific, often unknown environmental conditions in nature. Unlocking these silent pathways could lead to the discovery of entirely new classes of drugs or industrial compounds. The impact here is already substantial. Over 80 reports across 6 bacterial phyla already highlight this application. And recent advancements like mobile CRISPRi and Crage CRISPR are further facilitating the expansion of these tools into even more species, including hard-to-study pathogens and novel environmental microbes, even without fully sequenced genomes, literally unlocking a hidden chemical repertoire.
That's incredible, unlocking hidden potential. Beyond studying single genes, can they control more complex systems like entire genetic circuits or regulatory networks within a cell? It sounds like we could build new biological machines.
Absolutely, and this is where the precision and programmability of CRISPRa truly shine in the field of synthetic biology. These tools can be used to design and control synthetic transcription regulatory networks, essentially building custom genetic circuits within a cell, much like designing electronic circuits. It's remarkably effective because GRAs can be designed to target almost any sequence with the right PAM site, offering unparalleled programmability and modularity. You can combine multiple gRNAs, each targeting a different gene or regulatory element, to create complex logic gates like AND or OR gates, or feedback loops that respond to specific inputs.
Wow, cellular logic.
Right. These circuits can be fully synthetic, sort of auxiliary to the cell's native genetic regulatory networks. For example, they can act as heterologous sensors, designed to detect specific environmental inputs, maybe a pollutant, a biomarker for disease, or a nutrient concentration, and then trigger a programmed cellular response, like producing a therapeutic compound or changing cell morphology. They can function as multi-input circuits, processing complex information. Alternatively, CRISPRa can be interfaced with a host's native gene regulatory systems to control metabolism in response to internal stimuli, like cell density or specific metabolites, using either engineered or even the host's own sensor systems. This opens up possibilities for smart cellular factories. However, a word of caution here, it's a practical limitation. Expressing too many gRNAs simultaneously can lead to competition for the limited decast protein resource. This can potentially reduce the effectiveness of repression or activation on target genes. It's an important consideration for complex circuit design and needs careful optimization.
That's a great question. Let's break it down. So, what's an example of these networks in action, particularly in those challenging non-model bacteria you mentioned?
Well, while synthetic CRISPR regulatory networks are indeed less common in non-model bacteria compared to highly optimized model organisms like E. coli, there have been some pioneering studies showing it's feasible. For instance, a CRISPRa genetic circuit in Salmonella has been reported. This research showcased how you could implement a feedback loop in Salmonella, where the activation of a specific gene, mediated by CRISPRa, would then, in turn, regulate another gene, effectively creating a controllable switch within the pathogen. This demonstrates the potential for designing and implementing complex regulatory control, even in more challenging bacterial systems, enabling the cell to respond to and process information from its environment in a programmed way. The ability to dynamically control multiple genes simultaneously, often more easily and faster than traditional methods, makes these approaches incredibly promising for future biotech applications, from pathogen control to bioproduction.
Okay. Now, I've often heard about using CRISPR for metabolic engineering, which sounds like rewriting a cell's internal factory. How do these precise tools fit into that picture, especially for creating new, valuable products?
This is a hugely impactful area, particularly in industrial biotechnology and sustainable chemistry. CRISPR tools are being used to precisely redirect the flow of carbon and energy within non-model bacteria, essentially re-engineering their metabolic pathways. The ultimate goal is to shunt these biological resources towards the enhanced production of desired biochemicals, like advanced biofuels, bioplastics, or even complex pharmaceutical drugs and nutraceuticals.
Redirecting the flow.
Exactly. For example, CRISPRi is often employed to repress native genes, even essential ones, that might be competing for the same raw materials needed for your desired product, or genes that lead to undesirable byproducts. By subtly dialing down these competing or diverting pathways, you effectively push the carbon flux toward a specific product you want to synthesize. Imagine a bacterium that naturally consumes glucose for its own growth, but we want it to convert that glucose into ethanol. CRISPRi can dampen the bacterium's natural glucose-consuming pathways, forcing more glucose to be routed into the ethanol-producing pathway we've either introduced or enhanced. On the flip side, CRISPR can activate pathways to increase the biosynthesis of a desired product. Imagine a bacterium that naturally produces a valuable anti-cancer drug, but only in tiny amounts because the gene cluster for its production is weakly expressed, or silent, as we discussed earlier. CRISPR can be used to dramatically boost the expression of that silent gene cluster, leading to much higher yields of the drug. And here's the powerful insight. CRISPR isn't just turning a gene up, it's unlocking entire silent biochemical factories within bacteria that might have been dormant for eons. Think about it, we're talking about potentially discovering and exploiting novel antibiotics or complex biomaterials that evolution tucked away, now brought to light with a precision far beyond previous methods. This capability transforms drug discovery from a laborious, random search into a targeted activation, fundamentally altering how we might source future medicines and industrial compounds from the vast, untapped microbial world.
That's really profound. Activating hidden chemistry.
It really is. And these tools are often combined with other metabolic engineering techniques like deleting or overexpressing specific genes, or optimizing growth media to further increase the titers of the desired product. Some studies even cleverly use inducible systems to dynamically switch between cell growth phases and product biosynthesis phases within the same culture, optimizing resource allocation throughout the process. This allows for highly efficient and controllable production systems.
Okay, let's shift gears slightly. The ability to screen many genes at once, what we call high-throughput screening, is a monumental leap in scientific discovery. How do CRISPRa contribute to that, especially for large-scale studies looking at a whole genome?
You're absolutely right. High-throughput screening is a cornerstone of modern biological research, and CRISPRi and CRISPRa are excellent for conducting these large-scale screens, particularly for systematically investigating genotype-phenotype relationships. There are two primary formats, really. You can create pooled libraries where cells engineered with different gRNAs are mixed together, grown under selective conditions. Then the changes in gRNA representation in the surviving population are analyzed via next-gen sequencing to identify which perturbations influence the phenotype. This pooled format is more common due to its ease of use for very large genome-scale libraries, often exceeding 10,000 different designs. It makes it possible to screen every gene in an organism. We've seen this in studies with organisms like Staphylococcus aureus and Vibrio nitrogens, identifying essential genes or drug resistance genes.
So you analyze the survivors, basically.
Exactly. The alternative is arrayed libraries, where each gRNA design is constructed individually and cultured in a separate well, usually in microtiter plates. While pooled competitive selections are easier to scale to genome-wide dimensions, arrayed libraries allow for virtually any type of phenotypic analysis, since each perturbation is isolated and can be characterized in detail using microscopy, biochemical assays, or other high-content methods.
More detailed, but maybe harder to scale up.
Precisely. However, there are caveats with pooled assays. Because all cells directly compete within the same culture, cheaters can sometimes emerge due to complex competitive interactions. This means a gene's effect might be masked or misinterpreted in a mixed population. So individual gRNA results should ideally be verified in isolation to confirm their direct effect. Also, results from these pooled screens are sometimes specific to the gRNA design, not just the target gene itself, because of potential off-target effects where the gRNA binds to an unintended site. This could produce false positives or negatives. So, careful gRNA library design using sophisticated algorithms and empirical validation is absolutely critical. While genome-wide bacterial CRISPR screens haven't been as widely demonstrated yet as CRISPR screens medications, activation rules are a bit more complex. They're theoretically possible and definitely a promising future direction.
What do you think are single-cell RNA sequencing methods helping overcome some of these limitations in high-throughput screening, particularly with that cheater effect or the need for individual verification? It sounds like we could get a much richer picture.
Absolutely. Single-cell RNA sequencing is a truly transformative technology. It's a game changer here. It allows for high-content, unbiased transcriptomic analysis within pooled CRISPR screens, which is something we just couldn't do effectively before. The beauty of it is that each CRISPR perturbation and its resulting transcriptomic and phenotypic effects are confined to an individual cell. So its cells can be separated after the pooled selection, and single-cell RNA-seq allows for that meticulous separation and analysis of gene expression in thousands of individual cells. You can directly link specific CRISPR perturbations to unique transcriptomic readouts, and then to the observed phenotypic outcomes. This is critical for getting much deeper biological insights from large-scale screens, providing a far richer data set than just measuring overall fitness of a population. It helps deconvolve the complexities from competitive interactions in pooled assays by giving you a cell-by-cell understanding. It offers unparalleled resolution.
That's huge for interpreting those complex screen results. Okay, it sounds like there are immense possibilities with CRISPR and CRISPRi really pushing the boundaries of biology. But what are some of the practical hurdles researchers face, especially when trying to apply this to bacteria that aren't the well-studied lab rats, like E. coli or Bacillus subtilis? It seems like getting these systems to work in novel organisms would be a whole new level of complexity.
That's a great question, and yeah, it delves into the nitty-gritty of real-world research. Establishing CRISPRia in these non-model bacteria, which, let's be clear, includes the vast majority of diverse species out there, presents several significant and often interlinked challenges. First, there's the pervasive issue of domestication. This involves the often arduous process of figuring out the optimal conditions for culturing, maintaining, and reliably genetically manipulating a specific bacterial strain in the lab. For novel or fastidious species, those with very particular growth requirements, like strict anaerobes or obligate symbionts, it can be incredibly tough. It requires extensive trial and error and a lot of patience. Think of an obligate intracellular pathogen like Chlamydia trachomatis, notoriously difficult to grow and manipulate outside its host cell. Establishing suitable culture conditions for genetic manipulation can be a huge initial hurdle before you even get to CRISPR. This domestication process itself can take years.
Just getting them to grow reliably is step one.
Exactly. Second, many non-model bacteria are notoriously genetically recalcitrant, meaning introducing foreign DNA into them is exceedingly difficult. This is often due to their highly evolved native immunity systems designed to degrade foreign DNA. The most common are restriction modification, RM systems, which chew up unmethylated foreign DNA, and their own endogenous CRISPR systems, which target specific invading sequences. To bypass RM systems, researchers might need to painstakingly mimic the recipient strain's natural methylation patterns on the foreign DNA using specific enzymes, or engineer the recipient strain to delete or inactivate its RM systems. It's like trying to get past a sophisticated multi-layered security system without setting off alarms.
A lot of hoops to jump through.
Definitely. Third, reliable genetic parts, things like well-characterized promoters, ribosome binding sites, RBS, and terminators are often completely unavailable for new or less studied species. You can't just assume generic parts from E. coli will work effectively, or sometimes at all, in these different genetic contexts due to species-specific machinery. This means a lot of upfront work identifying and characterizing native regulatory elements within that specific bacterium. And to add another layer, expressing the DCAS protein itself can be cytotoxic if not tightly controlled, particularly in bacteria where high protein loads can stress the cell. So you need to find just the right balance, often requiring inducible promoters for tunable DCAS expression. Developing diverse libraries of genetic parts is crucial here.
Balancing act.
It is. Finally, avoiding interference with the bacterium's endogenous CRISPR systems is crucial. If your foreign CRISPRa tool, even a DCAS system, is too similar to the native active CRISPR-Cas system, it could trigger the host cell's own enzymes to cleave its own genome, leading to mutations or cell death. This means careful selection of the CRISPRa tool, ideally one that doesn't share significant homology, or one where the native system is inactive or disabled. You can predict these from sequenced genomes, but it's another hurdle. And even then, some bacteria have anti-CRISPR proteins that might need to be dealt with. It's a delicate balance, enough expression for regulation, but not so much it causes toxicity like changes in cell morphology or growth inhibition.
That paints a vivid picture of the sheer scientific ingenuity and persistence required. Now let's zoom out to the biggest picture, translating these technologies into human therapies. It seems like a monumental leap from fine-tuning bacteria to safely treating human diseases. What are the major challenges there for in vivo therapies, delivering these systems inside the body?
Yeah, for in vivo therapies, meaning delivering the CRISPR machinery and targeting genes directly within the human body, there are indeed significant technical and biological limitations that go way beyond what we discussed for bacteria. We're moving into patient safety and efficacy now.
Right.
The first and arguably most critical major hurdle is delivery. Getting the Cas9 or dCas9 machinery, whether it's DNA, RNA, or protein, to the specific target tissue or cells, effectively, safely, and only to those cells, is incredibly complex. While there have been remarkable advances, particularly with lipid nanoparticles or LNPs, showing promising results for delivering mRNA encoding Cas9 to specific tissues like muscle, brain, liver, and lung, it's not yet perfect. There's still often some degree of unintended uptake by non-targeted tissues, meaning more elaborate and precise systems for tissue-specific delivery are desperately needed. And methods for many other tissues, heart, kidneys, specific immune cells, still need development or significant optimization. This is a multi-billion-dollar problem in drug delivery, really.
The delivery problem looms large.
It really does. Then there's a looming issue of unwanted immune responses. Our bodies have sophisticated immune systems to fight off foreign invaders, and unfortunately, Cas proteins being derived from bacteria can be recognized as foreign. Studies on pre-existing immunity to common Cas proteins, like Streptococcus pyogenes Cas9, SvCas9, in humans have shown conflicting results, but generally suggest it's a significant factor. Some reports show up to 58% prevalence of antibodies or T-cell responses, others lower figures. But the consensus is it's a critical consideration. This immune response could lead to rejection of the therapy, reduced efficacy, or even adverse reactions. That's why researchers are actively searching for new Cas orthologs like Staphylococcus aureus Cas9, SAC-Cas9, which often show lower pre-existing immunity.
Our own defenses working against the therapy.
Potentially, yes. Furthermore, off-target effects remain a persistent concern for all CRISPR-based technologies, especially in therapeutic applications where absolute precision is paramount. While DCAS9 and its effectors are generally more precise in binding than conventional Cas9 cutting, they can still bind to unintended similar sequences, particularly in overlapping promoter regions. This could potentially interfere with the expression of neighboring, non-target genes, leading to unforeseen consequences. For therapies, this needs to be minimized to virtually zero. So precision controls like inducible systems, inhibitors, degraders to rapidly switch off Cas9 or DCAS9 are actively being developed as a safety switch. Also, for therapies still relying on conventional Cas9, the double-strand breaks it creates can induce DNA damage responses, which in some cell types can lead to low editing efficiencies, chromosome rearrangements, or even cell death. These are all critical safety hurdles.
So, given these profound challenges, what's being done to overcome them? Are there completely new tools or strategies on the horizon that are making these precise gene modulation techniques more feasible for both basic research and, critically, for human therapeutics? It feels like the field is moving at warp speed.
It truly is a dynamic field, and researchers are innovating at an incredible pace. A major leap forward tackling many of those issues has been the development of base editors and prime editors. These are next-gen CRISPR tools allowing for even finer, less invasive modifications.
Okay, base and prime editing.
Exactly. Base editors, for example, are a significant advancement. Unlike traditional Cas9, they don't create double-strand breaks. Instead, they consist of a dCas9 or a Cas9 nickase, which only cuts one DNA strand fused to a DNA deaminase enzyme. These deaminases chemically convert one specific base into another. So you have cytosine base editors, CBEs for C to T conversions, and adenine base editors, ABEs for A to G conversions. This is huge because it dramatically reduces cytotoxicity and the unwanted insertions or deletions, the indels, that come from repairing double-strand breaks. They perform a precise chemical search and replace on a single nucleotide.
Chemical editing, not cutting.
Precisely. These base editors have already shown incredibly promising preclinical results in animal models. For instance, in Hutchinson-Gilford progeria, a devastating premature aging disorder caused by a specific C to T mutation, base editors successfully corrected this in vivo, extending mouse lifespan by a remarkable 78%. They're also being explored for high cholesterol by targeting the PCSK9 gene, with promising results in primates showing substantial, durable cholesterol reduction, with no major off-target editing or long-term immune issues reported so far. There are even new dual deaminase-based editors doing both A and C editing.
Wow, real therapeutic potential showing up.
Definitely. However, base editors aren't without their own limitations. They can still cause some transcriptome-wide off-target RNA editing, which researchers are working hard to minimize by developing new deaminase variants or refining the Cas9 editor relationship. And critically, current base editors can't perform all 12 possible base-to-base modifications, so they can't correct every single point mutation.
Okay, so still some limitations.
Which led to the development of prime editing, which is even more versatile and addresses many of those remaining gaps. Prime editing uses a Cas9 nickase fused to a reverse transcriptase enzyme, along with a special prime editing guide RNA, or PEG-RNA. This PEG-RNA not only guides the nickase to the target, but also provides the template for the new DNA sequence to be written in. This allows for search and replace genome editing, enabling all 12 base-to-base conversions, plus small insertions and deletions, all without double-strand breaks or needing separate donor DNA templates. It generally has even lower off-target rates than conventional Cas9 and offers cleaner, more versatile DNA modification. The main limitation right now for prime editors is often lower efficiency compared to base editors for some edits, and they struggle with inserting very long sequences efficiently. But it's an incredibly powerful leap in precision.
So even more refined control.
Absolutely. Looking ahead for bacterial applications, we definitely need stronger and more versatile CRISPR tools. This might involve engineering new Cas systems with relaxed PAM requirements, allowing targeting of more sites, or directed evolution of activator domains for more potency. For therapeutics, the focus is squarely on optimizing base and prime editors for higher efficiency and fewer off-targets, improving those crucial delivery systems for tissue specificity, and developing tailor-made gRNAs based on a patient's unique genetic makeup. This personalization is key. The rise of translational dCas9-based technologies that allow precise dose and temporal control of therapeutic targets without creating double-strand breaks is also a very exciting avenue. It sidesteps many Cas9 challenges by just modulating gene expression, offering promise for dynamic, reversible, and potentially safer therapies.
Wow, what an absolutely incredible deep dive into the fascinating world of gene regulation. So to recap, We started by learning that CRISPRa and CRISPRa are like incredibly sophisticated molecular dimmer switches for our genes. They leverage a modified dead version of Cas9 called dCas9, which can precisely locate specific DNA sequences thanks to its gRNA, but crucially, it cannot cut the DNA. Instead, this dCas9 acts as a highly targeted delivery system, carrying either activator or repressor tags to turn gene expression dynamically up or down.
And we saw how these ingenious tools overcome many of the limitations of older technologies like RNA interference and even traditional CRISPR knockout, offering more precise, titratable, and often less toxic control over gene expression. They are truly revolutionizing how we map gene function on a large scale, build complex, programmable genetic circuits within cells, engineer microbial metabolism for the production of valuable biochemicals, and conduct incredibly powerful, high-throughput screens to discover new biological insights.
Yeah, absolutely. And despite the significant challenges that remain, particularly concerning delivery, potential immune responses, and minimizing off-target effects, especially in human therapies, the field is moving at an astonishing pace. Groundbreaking innovations like base editors and prime editors are paving the way for incredibly precise genetic fine-tuning without the double-edged sword of DNA breaks, offering a new paradigm for both research and future medicine.
If we connect this to the bigger picture and really let our imaginations run wild for a second, imagine a future where instead of one-size-fits-all drug treatments, we could personalize therapies not just to an individual's unique genetic makeup, but to dynamically control gene expression in real time within specific tissues or even individual cells without ever permanently altering their core DNA. This raises an important question, doesn't it? How will this unprecedented level of fine-grained cellular control fundamentally reshape our understanding of health, disease, and maybe even the very definition of human potential? What new biological frontiers will we cross when we can truly dial in the instructions of life?
An incredible thought to ponder and a powerful vision for the future. Thank you for joining us for this deep dive into the fascinating world of CRISPRa and CRISPRi, the precision tools that are truly transforming biotechnology and medicine before our eyes. Thank you for joining again.